Hello everyone, welcome back to the second module of the lecture series. So in the first module we have discussed about the prokaryotic DNA replication, and in that video we have discussed the semi-conservative model of DNA replication, whereas we also discussed the different enzymes and protein involved in DNA replication process. So let's discuss about the initiation, elongation, and termination process in this video series. So let's start with the mechanism of DNA replication process. So DNA replication have three main stages. One is initiation of DNA replication. In initiation, first different proteins and enzymes will be recognize the uh, origin of replication site of DNA replication. As we know, the prokaryotic origin of replication have only one origin of replication site, and it is the length is about 245 base pair in length, and it has different sequences. And within that sequence, the different proteins or helicase protein will be bound, and it will start melting the DNA. So in initiation, we can subdivide the stages into two different steps. First is your initiation or origin unwinding, and second one is your replizo assembly. And the second step is the after initiation, there will be the elongation. That means the polymerase, DNA polymerase 3 will synthesize both leading and lagging strand. Then third step is the termination. In termination, the DNA replication has to be stopped in a particular point where the parent strand and the newly synthesized strand will be released from the system. So this is all about the overall three stages of DNA replication. Initiation, where that occurs at the origin of replication site, where the double-stranded DNA will be separated, they will form replication fork, and primer will be synthesized. Then after, in elongation step, there will be addition of DNTPs or new nucleotides to the complementary strand, and with respect to the template strand. So there will be also the formation of phosphodiester bond, and if mismatch will be there, that will be rectified and strand will be elongated. In termination, there should be the stop in DNA replication process at the termination site. So starting with the origin recognition or origin unwinding process, as prokaryotic DNA origin of replication have different segments or different sites. So at this origin of replication in the picture, we can see starting from the R1 means this origin contain three directional repeats that are 13 nucleotide in length, three directional repeats of 13 nucleotide length sequence. It is called as DUE region. DUE stands for DNA unwinding elements. In the proximity of the DUE region, there is a segment that is DNA triose, as you can see here. Following the triose, we have a region called R1, R2, R3, and R4, which are 9 nucleotide in length. In between the region, there are other 9 nucleotide long sequence which differ slightly from the main region 1, 2, 3, and 4. Next to R1, there is a sequence which specifically recruits a protein called integration host factor, IHF. One last thing to be noted, that the region on the left of the R2 are directionally oriented to towards the DUE region. The region from the R2 till the R4 are oriented away from the DUE region, and Starting from the R1 until the R4, this entire section is called DNA A assembly region. So the main part of the DNA of the origin are these LMR repeats in the DUV region, then the triose region and the DAR site. The second main thing that we need to recall is the DNA A initiator protein. These in the second picture you can see the DNA A initiator protein, and it has four important domains. Domain 1 forms oligomers, domain 2 is a flexible linker, domain 3 can bind single stranded DNA, and it is also an ATPase activity domain, and finally domain 4 binds the double stranded DNA. Specifically, domain 4 binds the high affinity region 1, 2, and 4 in the DAR site, and domain 3 is not really a high affinity site. Domain 4 also bind to the internal region that not as readily as 1, 2, and 4. Okay, so let's start discussing two model of the origin unwinding. There are two main models to discuss. One is the two-state model and other is the loop back model. Okay, the two-state model suggests that there are two different DNA A oligomerization state. What DNA A oligomerization state means? In the final step of the origin unwinding, there is a DNA A oligomer present at the DAR region, which is associated with the double-stranded DNA. And this is the first state. And then there is another DNA A oligomerization that occurs at the DUE region, which occurs in the context of single-stranded DNA. And this is the second step. In contrast, according to the loopback model, there is a single DNA oligomerization state, but it invokes the folding or loop of the DUV elements. You can see at the picture. That means there is a DNA oligomer that assembles at the DAR site, and when the origin unwinds at the DUV folds back onto the DNA A oligomers at the DAR site. So there is a good amount of evidence for both of these models. The usage of this model probably depends on the context of protein concentration, origin structure, and a bunch of things, other proteins. In this, uh, in these two models, let's start discussing individual model in details. The focus of origin begin in the two-state model is with R1, R2, and R4 region because they are high affinity region site of the DNA A protein. The DNA A domain 4 recognize the region and bind to the particular site, which means that in the picture the fastest domain is the domain 1 and the red color is the domain 3. And more specifically, the DNA A ATP binds better at this site, but ADP forms works as well. So the first step, the DNA A, for the first step is the binding of DNA A at the high affinity region of the DAR. Then DNA A protein actually comes with the helper protein, which is called DIA. DIA in either come in the in DNA DIA precomplex form, and it can bind to a DIA after it will bound to DAR in order of the event with the helper protein. In this case, it's not, by the way, the DIA stands for DNA A initiator associated protein. So now we have to we have to discuss the DIA. Well, in turns, DIA can form tetramers, and if the DIA can form multimers, then also form it can also form multimers, since DIA is bound to the domain one of the DNA. This sort of association is called the linkage effect of the DIA, and this drives the DNA oligomerization. So since multiple DIA can come together, it can force the aggregation of DNA protein at the DAR site, and it is likely to the crowding agent. Once this DIA forms multimer, it can further force the DNA oligomerization at the DAR site. So in the DAR site that are low affinity site for the DNA, once this DIA mediated linkage is forced, more DNA is recruited at the DAR site, and it occupies the low affinity sites. And these new DNA and more these new DNA protein also have DIA associated with them, and so further stabilizes the DNA complexes at the DAR site. This mechanism of DNA association is probably concentration driven, and this binding of DNA complex at the DAR also correlates with the binding of IHF at the IHF recruiting sequence near the region 1. And this completes the state 1 of the 2 state. And this binding of DNA complexes at the DAR also correlates with the binding of IHF at the uh, IHF recruiting sequence 1. Uh, the transition of the next state depends on the IHF, which assists in the DNA unwind of the DUV region, which is just upstream of the R1 site. The unwinding or DNA melting is caused because IHF bind IHF binding causes a 120-degree bend in the DNA, and the unwinding then allows DNA oligomerization into the DUV region. And this is the second state. Let's start discussing the processing visually. So, by emphasizing the binding of IHF, we cause the DNA distortion, uh, distortion, uh, which is a fancy word we can say for change in the DNA topology. All these change occur in the backdrop of a stable state 1, which consists of DNA stably bound to DAR region at both low and high affinity site. This state 1 is also stabilized by DIA because DAR is stably a stable state 1. And dairy is stably bound to DIA protein, and it is hard to unwind the DAR. So the IHF mediated topological change bound by DNA protein, it is hard to unwind the DAR. So the IHF mediated topology, um, since unwind the DV region, and it is bound at the any protein. It is not bound at any protein, and also at DV is very AT rich, so it is easier to unwind. So then second region is the trios that overlap a little bit with the DV. So the trios and the LMN repeats become exposed at strands of DNA after unwind, and these become the target of binding of DNA ATP molecules. By the domain, in contrast, that region are bound by DNA domain 4. So in this picture of single stranded DNA binding at DUV, DIA may away from the complex, as we will discuss the mechanism of DIA dissociation in later in coming session. The dissociation enables a steady state transformation, which is in forward equilibrium, to move into the replisome assembly state. To understand what is happening in this, we have to noted that the domain 1 of the DNA A can form an oligomer with another DNA A domain 1, so it can dimerize. So once DIA is gone, the domain 1 of this DNA A domain 1, uh, so it can dimerize, and domain 1 of the DNA A protein can interact with each other. This is highly dynamic state, which is possible because of the flexible linker domain 2. One last thing, the domain 3 interact with the LMNR motif specifically on the T-reach part. The assembly of DNA A on the origin completes the second state of the two-state model, which has a stable single standard DNA protein complex, whereas the first state is the DAR region in the double standard DNA protein complex. This is how the two-state model position origin unwinding happens. To the recruitment of helicase and other protein needed for the replisome assembly and continuation with the DNA replication process. Now let's discuss the loop back model. The initiation of the loop back model is exactly the same as the state 1 of the and state 2 state 1 of the state 2 model, which is until this topological model is exactly the same as the state 1 changes state of the origin, leading to DUV unwinding. For clarity, let's start discussing the thing in a proper way. So, leading to DUV unwinding, the DUV is unwound and the DNA A protein do the things, and not all. In the loopback model, the DNA A protein do not oligomerize into the DUE region. This is in contrast to the two-state model where where oligomerization proceed from the triose into the DUE region. In the case of the loopback model, it doesn't happen. The melted DUE region folds back. This means the single standard from the DUV contra, the DA which is present and on the DAR side, and this contact means that domain 3 of this DNA are associated with the DUV region while it is bound to the DAR at the same time. This is once again possible because of the topological change created by IHF protein. This leads to the formation formation of a stable single standard DNA and protein complex, which is ready to recruit helicase. Before we end here, I want to point out some of the amino acid involved in this interaction. The double standard DNA binding of DNA A domain 4 is occurring through a helix-turn helix domain, which contain arginine residues that scan the major and minor group of the 9-mer repeats. Then domain 1 oligomerization and sometimes domain 3 head-to-tail oligomerization involves the interaction of the arginine residue. Domain 3 of DNA A protein binds the single standard DNA at the DUE region because of specific arginine as well as valine residue. So arginine seems to be a very crucial amino acid to mediate this interaction that And the second issue related to the fundamental observation that primase and helicase complexes. So next, we will discuss the replisome uh, assembly. This is all about the replisome assembly and how DNA C, DNA B, which is the helicase, are involved and assemble, and DNA G, which is the primase, is involved. The DNA A protein on the origin uh, uh, DNA A is after unwinding the DNA, matlab, sorry, now following this origin unwinding, the DNA A is released, or rather it has to release. Although the exact mechanism for its release is unclear, people have uh, um, speculated that there might be a DIA release factor, but it is generally not known. Sometimes this job of DIA is also done by another set of protein called HU protein, and but the point remains that DIA has to be released after the origin opening. Now you may ask, why do DIA protein need to dissociate away? After all, it stabilizes the DNA on the origin, right? So the fact that the binding of DIA on the DNA protein comes on the domain 1, specifically at the phenylalanine at position 46. Now the transition into the replisome assembly, we start by helicase loading, actually depends on whether or not phenylalanine 46 is available or not. So it needs to be accessible. But if DIA is bound to that amino acid, then it is not clear, and therefore DIA needs to be released. So from the DIA protein needs to be released from the origin. Now we get into the then helicase, also known as DNA B in E. coli, loading depends on the loader protein called DNA C. Helicase, this idea is similar to that of loading RNA polymerase at the promoter, which in prokaryotes depends on the sigma factors, and TF2F in case of eukaryotes. Depends on some people actually also say that DNAC is not actually a loader protein, given that it never actually loads. It just increases the frequency of loading at the origin. But for the sake of simplicity, we will stay away from the discussion and call DNAC is a loader. The active form of helicase is hexamer. You can see the picture in the bottom, and meaning it is formed by 6 monomers, and the whole complex is stabilized by magnesium ion. A monomer of the helicase may look sometimes like different, and it has n terminal and C terminal end. The N terminal is smaller end, and C terminal is a bigger end. So you have this back end N terminal and the front end C terminal. This hexamer looks like a cylinder, and the internal channel is positively charged, which is convenient because DNA is negatively charged. So it has some nice ionic interaction between them. So the smaller C terminal domain is the one which has the helicase activity, and it also has the ATPase subunit to power the helicase forward. To load the helicase, DNA-C interact with the domain of domain 1 of the DNA-A protein, which also already at the origin, and the loader protein also interact with the domain 3 of the DNA-A. The additional help in the loading of helicase comes from the helicase itself. The N-terminal domain of the helicase interact with the phenylalanine 46 in the DNA-A domain 1. This is the same amino acid at which the DNA A was bound. The N-terminal domain of the helicase interact with the phenylalanine 46 in the DNA domain 1. So, to add some details, the loader protein, about the loader protein, you should note that the DNA C binds to the C terminal end of the helicase, and usually three of them binds to the hexamers. Now the DNA C protein interact with the DNA A domain 1 and 3 and loads the helicase at DUV region, where the DNA C is bound to the C terminal of the helicase. Notice the positioning of the N terminal and C terminal of the helicase. The strand is 5' to 3'. As we know, this 5' side has N terminal region, whereas the C terminal region is facing towards the 3' end. And we have said before, you will find that there are three loader proteins present on the hexamer. So the ratio of molecules is loader to helicase is 3 to 6. The 6 comes from the fact that the helicase is hexamer. Note that this top and note that Note that this top strand goes from 5' to 3' and then there is another 5' to 3' strand in the anti-parallel direction. And given the loading direction of the helicase, we can draw the outline. We can see the outline of the helicase on the on this bottom. And N-terminal domain is at the 5' site and the C-terminal domain is at the 3' site. So essentially, this means that the loading of helicase on this top strand is mediated primarily by the DNA-C, DNA-A interaction, whereas the loading of the bottom strand is mediated Whereas loading of bottom strand is mediated by N-terminal domain interaction with the domain 1 of DNA-A and specifically by the phenylalanine interactions. So this doesn't mean that the N-terminal of the other helical does not interact with the DNA-A, but its position suggests that interaction in the other cases is less frequent. And like the DNA-C position on this helicase means that its interaction with DNA-A is less frequent. So to the sum up, we have the DNA-B N-terminal interface and DNA-C faces the mediate the and DNA-C interface that mediate that some aspect of helicase structure. And N-terminal and C-terminal views, you can see it here. And C-terminal view is straightforward if it contains fairly homologous hexameric organization that looks like cylinder and it has central channel which is open. N-terminal view, on the other hand, reveal a very different story. It has this structure that looks like dimer that comes together as a trimer. As each of these dimer has this notch kind of structure, to move to the next step, I will simplify this thing, only use the top strand of the explanation. So far, we have discussed DNA-C and DNA-B organized on the origin, where DNA passed through the positively charged channel of the helicase. In recap, as we know, the helicase moves in the 5' to 3' direction and its helicase activity comes from the C terminal domain. However, currently the DNA-C protein has occupied the DNA-C terminal domain of the helicase, and this essentially blocks the helicase and ATPase activity of the DNA-B hexamer. The loader protein of the helicase is also its negative regulator. So if a helicase has to move ahead, the loader protein must be released. And this release occurred due to the recruitment of DNA-G, which is a primase protein that uh, primase protein that uh, all of you have know. Uh, so So in simple term, as we can call it as a recruitment of DNA G kicked out DNA C of DNA B. And for your reference, the N-terminal domain in the other end of the helicase, which is DNA C, is bound to the C-terminal end. Simply speaking, the binding of DNA G at the N-terminal region causes its conformational changes that activate the ATPase in the DNAC. And by the way, DNAC has the ATPase domain as well, and the ATP hydrolysis causes the loaded protein to be released from the helicase. But how is this conformational change is occurring? So where the N-terminal view comes in handy, it turns that DNAG binds at these notches at the N-terminal domain of the helicase, and this is what it means. DNAG is loaded to helicase, and this binding of primase at the N-terminal region locks the helicase position so that it engages the DNA in the central channel much better. This lock causes the loader protein to be released. Interestingly, here also there are three DNAG proteins are the DNAG proteins for the entire helicase, the same ratio as we saw with the DNAC. In this view, DNAC is released and the primase is bound to the N-terminal region, which makes primer. This brings us to the next problem, which is in the incompatibility in the primase. And to understand that, we have to know that DNAG is basically a specific RNA primer, RNA polymerase. So it uses DNA and makes RNA. So now let's define the issue of incompatibility with the helicase. So as we have noted so far, the helicase moves from 5' to 3' direction, and how DNA-G makes RNA and synthesis direction is also 5' to 3', which means it movement on the template must be from the 3' to 5'. But the movement of helicase on the template is 5' to 3', and so the helicase wants to move forward, but primase, which is physically connected to the helicase. Hence the problem that DNA-G and DNA-B are active in the positioning direction. Hence the so how does this actually works? So, but it is not a problem for the cell. To understand this, let's discuss how it works. So, from the side, this will look something like this, where DNA-G is connected to the N-terminal domain of the DNA-B, and the single-stranded DNA runs from the channel of the helicase domain of the DNA-B. And the single-stranded DNA runs from the channel of the helicase. The helicase is moving towards 3' end. The active site of the primase, which contain the RNA polymerase domain, is at the distal end. And before the DNA gets to the RNA polymerase site, the single-stranded DNA is looped into, and this loop because this active site tries to recognize the priming site, which is specifically the CTG on the single-stranded DNA. And it has the high affinity recognition for the primase to start making the primer. And the first nucleotide to be synthesized is the adenine and the guanine, and it moves towards the 3' end. And the G is only used for the recognition and not as a template. The primase wants to synthesize the primer towards the 5' direction on the template, but the helicase is moving towards the opposite direction. Now comes the solution. What actually occurs here is After that, the CTG recognition, the DNA is not allowed to pass through the primase and the synthesis begins on the template. But since helicases keep moving, a loop of single-stranded DNA comes out in between the primase and helicase, and the primer synthesis can still occur. Just keep in mind that the primer will also uracil in it, given that it is a RNA polymerase. That is, this sort loop is stabilized by some single-stranded binding protein. So this is how the recognition priming site by DNA G and helicase movement are coordinated. This is also means that the RNA primers are relatively short and they ranges from 10 to 60 nucleotide, but 90% of the time they are only about 11 to 12 nucleotide in length. If you have long primer, the size of the single-stranded DNA loop between primase and helicase will get too big and that will destabilize the structure. And this is a good transition of this state. And what exactly determines the length of the primer? It is a question. And to answer this, let's start discuss the structure of the helicase complex. Helicase, as we know, is a hexamer. There are usually three primases bound to the N-terminal region, and out of three, only one primase is actually function to make the primer. And the this active DNA G, here is a loop that comes out between the primase and helicase. And so one control of primer length comes from the fact that the flexible loop cannot get, if it doesn't, then the loop and the corresponding RNA duplex will be will be destabilize the active primase and it release. The other mechanism relies on the other primase. The primases are also pretty flexible, and there are some studies that show the mere dangling of these non-active primases will interface with the active primase and cause its release. And this also appeared to be the region for the control of the primer length. Now, there is actually one interesting fact that relates to the DNA polymerase activity in the bacteria, which fits with the short primer length. And if you know how the RNA primer length and DNA polymerase activities replication are related, then in the next um, then now the regulation, the active primase is release, and sometimes all primase release from the helicase, but usually one or two stay bound. Now in this complex, we have two and the RNA primer, which is left behind. On the another fact, the length of the DNA in the helicase is around 20 nucleotides. And here is one last thing before we wrap up the thing. When the primase is actively making RNA, the helicase is moving the DNA B movement. Helicase is moving and the DNA B movement is actually pretty slow. But when the active primase is released, the helicase is actually faster. This is not surprisingly to imagine because of the immediate single-stranded loop that appears between primase and helicase. This is also so that primase and helicase are indeed a bit incompatible when it comes to optimal speed of the helicase movement. So I hope that helps you to understand some key ideas about the origin unwinding process. We have the DNA G, DNA B complex, and on one strand position accordingly. So this positioning of the top complex occurs at the L region of the DUE, and this and the position bottom and the positioning of the bottom complex occurs at the R region of the DUE, which partially overlap with the triose. This is the activated state of the helicase where an RNA primer has been made, and the helicase can actually move and unwind the genome starting at the origin. The actually unwinding process will be discussed, and we will start discussing the unwinding process in the next module. Okay, thank you so much.